Hey everyone, welcome to This Explainer. Today, we're taking a fascinating journey inside what is literally one of the most efficient, hyper-coordinated assembly lines in the known universe. I'm talking about your very own cellular factory. We're going to explore the kinetic mechanics of how your body actually translates a genetic blueprint into the physical proteins that make you, well, you.
It's a really profound question, isn't it? How does a cell read a microscopic genetic blueprint and physically construct the machinery of life? Well, today we're breaking down this highly coordinated biological assembly line known as protein translation. We're going to look at the specific machinery, the exact sequence of events, and those remarkable finishing touches that turn simple chemical codes into a functional reality. Okay, let's dive right into this. To make sure we capture all the amazing details, here is our roadmap. Section 1 is the genetic blueprint, followed by translation components explained. Then we'll hit the active stages: initiation, elongation, and termination. Finally, we'll wrap up with Section 6, post-translational modifications. All right, Section 1, the genetic blueprint. So, before our factory can actually build anything, we have to understand the language it's reading, right? And that language is messenger RNA. Because mRNA is made of 4 different nucleotides, there are exactly 64 possible trinucleotide sequences available to code for our 20 amino acids. Notice AUG? That is your universal start signal. It codes for methionine and basically initiates the whole chain. Then you have things like UUU, which gives us phenylalanine, a structural building block. But importantly, down at the bottom there, UAA, UAG, and UGA, those don't represent any amino acid at all. They act strictly as stop signals. Which brings us to a really crucial concept, the codon. A codon is simply a sequence of 3 consecutive bases on an mRNA molecule that forms a single word in this genetic code, representing one specific amino acid. And because we have 64 possible codons for only 20 amino acids, well, do the math. Most amino acids are actually coded for by more than one codon. It makes the genetic code beautifully redundant, or degenerate. Now, why does that redundancy matter? Well, it creates something known as the wobble effect. The wobble occurs at the 3' end of most codons, and it essentially enables non-standard base pairing between the codon and the delivery molecule, the tRNA. This means one single tRNA can interact with more than one codon. It's basically nature's brilliant biological failsafe against minor genetic mutations.
Moving on to Section 2, Translation Components Explained.
Now that we understand the language, let's meet the cast of characters working on the factory floor. You've got your mRNA blueprint, your functional ribosomes acting as the main assembly machinery, and your energy sources, ATP and GTP. But I want you to pay special attention to those tRNAs. They're like the adapter delivery trucks, and a specific enzyme called aminoacyl tRNA synthetase. This enzyme is vital. It ensures the correct amino acid is loaded right onto the CCA sequence at the 3' end of its specific cognate tRNA, and it activates it for peptide bond formation.
Let's look a little closer at that main assembly machinery, the eukaryotic ribosome. It's known as an 80S ribosome, and it's actually composed of 2 distinct subunits. You have the 40S smaller subunit, which contains 18S RNA and an incredible 33 distinct proteins. And then the larger 60S subunit is even more staggering. It holds 5S, 28S, and 5.8S RNA, alongside 49 proteins. Together, these 2 insanely dense, complex subunits lock together to form the functional 80S factory site.
Which brings us to Section 3, Initiation, starting the engine.
Okay, so with all our machinery on the floor, the engine starts. Initiation is literally like firing up the assembly line, and it happens in 4 distinct stages. First, initiation factors bind directly to the small 40S subunit. Second, a ternary complex, which includes the unique initiator tRNA carrying methionine, joins in to form the 40S pre-initiation complex. Third, the mRNA binds, using its 5' methyl cap almost like a guide rail, and it scans along for that precise AUG start codon, which is located at a specific sequence called the Kozak consensus. Finally, the large 60S subunit locks in. When this is all complete, the initiator tRNA is sitting perfectly in what's called the P site of the ribosome, leaving the adjacent A site wide open and ready for business.
Let's move to Section 4, Elongation, growing the chain.
So the A site is open, which means it is time to build. Elongation is the fastest, most repetitive part of the assembly line, just a continuous cycle of 3 main events. Step 1, a new incoming aminoacyl tRNA binds to that empty A site dictated perfectly by the codon that's currently exposed. Step 2, a peptide bond is formed between the new amino acid and the growing chain. And step 3, translocation. This is a physical shift where elongation factor EEF2 and GTP power the movement of the ribosome exactly one codon down the mRNA. This shifts the newly bound tRNA into the P site, completely freeing up the A site for the next cycle.
Watching this continuous kinetic flow back and forth between the P site and the A site inside the ribosome is honestly mesmerizing. But what's also amazing is the sheer efficiency of it all. Because mRNA molecules are so long, more than one ribosome can actually translate the exact same mRNA simultaneously. This complex is called a polyribosome, or polysome, and it maximizes the factory's output tremendously.
Actually, before we leave elongation, I have to point out a truly surprising fact about step 2. The peptidyltransferase that catalyzes that peptide bond, it isn't a standard protein enzyme. According to the source material, it's actually the 23S rRNA component of the 60S large subunit. That makes it a ribozyme, an RNA molecule literally acting as an enzyme. It's a mind-bending piece of biological architecture.
All right, Section 5, Termination, stopping the assembly. Eventually, our chain reaches its proper length, and the factory needs to know exactly when to stop building. Remember those 3 specific stop codons we looked at earlier, UAA, UAG, and UGA? Termination occurs exactly when one of those terminator codons slides right into the A site. Because there's no tRNA with an anticodon that recognizes these stop signals, the whole process halts. Hydrolysis occurs, which essentially severs the bond between the peptide and the tRNA occupying the P site, and that successfully releases the brand new polypeptide chain off the assembly line and out into the cell.
And that brings us to Section 6, post-translational protein modifications.
So the assembly line might be stopped, but the raw protein product still needs a few finishing touches before it can actually function in your body. We call these covalent modifications. They can include the removal of that initial methionine or the cleavage of translated sequences, which, by the way, is exactly what happens with pancreatic islet cell hormones. You also have glycosylation, which is adding sugars from membrane proteins, or phosphorylation. And here's a great example of why this matters for your everyday health. Certain structural proteins require vitamin C-dependent hydroxylations right at this cellular stage just to properly build functional collagen in your skin and joints. No vitamin C, no collagen.
The final step is folding. The raw chain has to fold into a very specific, functional, three-dimensional native structure. This process usually begins while the chain is still being synthesized, and it's assisted by molecular chaperones, like heat shock proteins. They act as active catalysts, guiding the protein into its proper 3D shape by actively inhibiting improper attachments and kind of pulling apart unwanted liaisons.
Which brings us to the end of our explainer. From the moment the mRNA blueprint is read, through the kinetic dance of the tRNAs and the ribosomes, right down to the final folding by those molecular chaperones, it's just a flawless, nonstop cellular factory working endlessly to keep you alive. But it does leave you wondering, with millions of these microscopic assembly lines running every single second, what exactly happens when the machinery makes a mistake? Well, something to think about.